All right, thanks very much. So my name is Lou Madsen. I'm in the chemistry department, and my group predominantly does magnetic resonance studies on ion conductors and drug delivery agents. I'm sort of an NMR person with an NMR background, but I'm not an NMR purist in that I do sort of solids or liquids or imaging. I pick what techniques work to solve specific problems and use techniques that make sense. Okay, so all right. So the title of this project, which we're undergoing with Judy Riffle, also in chemistry and test, is transport properties of sensitive nanocluster MRI contrast agents. So essentially, Professor Riffle's group is making a wide array of nanoclusters of magnetic particles, and they're held together and coated with polymers. Okay, and these can be tailored. The polymer structure here and the size of the nanoparticles and the size of the clusters can be tailored both to carry drugs and also to allow for imaging capability. Okay, so if we just look for a minute at one little iron oxide nanoparticle. Okay, iron oxide is in a health sense benign, right? It's basically something you could eat. Okay, and so basically you get an MRI imaging contrast by this emitting a magnetic field. So you have a spatially varying magnetic field. Water comes in and out of this magnetic field, and the water is what you're measuring in magnetic resonance imaging. And so it changes the contrast in your magnetic resonance image if you have water circulating around these particles. If you put these particles into clusters, you can actually further tailor the degree of that water signal change in the image. So these clusters, which Ripple's been working on for quite a while, you can tailor the size of these clusters to get both T1 and T2 contrast. So if you're familiar with MRI, there's spin-spin relaxation T2 and spin-spin relaxation T1. You can adjust those things by changing the sizes of these nanoclusters and how the clusters are held together. And these actually have some of the highest T2 relaxivities, I guess maybe the highest ever measured. And you can also load these with drugs. So what we've been trying to understand is, what we're trying to understand in this project is, how do nanoparticles like these transport through tissue? So if you're familiar with drug delivery literature, typically people come up with a drug delivery vector and they test it on a cell. Okay, they look for cell transfection or cell uptake. And then sometimes they can tether on a contrast or imaging agent and make a theranostic particle out of that. Okay. Previous studies in these areas basically address two extrema. One is the particles going into cells. So can we transfect cells? Can we treat cells with these particles in vitro? So you basically make a cell pack, you put your nanoparticles in there, and see if they go into the cells, and if they kill the cells or if they produce the desired effect in the cells. And then the next thing that people do if they pass the cell test is they do in vivo tests, right? They take a mouse or a rat and they do tail vein injection, and they see where the particles go. This is a sequence of magnetic resonance images of mice where you have these particles going into the liver. It's kind of a small image, but after some time, these particles rapidly locate into the liver. Okay, and they turn the liver from brighter to darker. Okay, these are these actual nanoparticles. So I look at this problem and I say there's plenty of room in the middle. Okay, basically we can understand what happens to the cells, we can understand what happens to an organism, but what happens on the tissue level? How do these particles transport through tissue? When you get in arteries, fine, that's easy to understand. But once you get to small capillaries, or say intrices, pathways between cells, that's unknown territory. As far as we can find in the literature, there's basically no studies addressing this problem. So we're basically trying to use. Okay, so I'll get to that. How we're going to address that in a minute. The team is composed in my group of Xiaoling Wang, who's in the back, and another student, also been joining this project to do some time-resolved magnetic resonance imaging transport studies. And then the Riffles group, Napon Poti, is doing synthesis and engineering of these particles. Okay, so how are we going to do this? Basically, we can use the intensity in a magnetic resonance image to track the transport of these particles. So essentially, we're doing time-resolved imaging, and we're looking at how the intensity in the image changes spatially. Okay, and we can use that to get things like the diffusion coefficient of the clusters, or if we did something like applying a pressure or a flow gradient in the system, or some other, okay, we heard a great talk yesterday by Carlos Rinaldi, who was doing magnetic guided therapy, thermal therapy. You can use magnetic fields to guide the delivery of particles. We can basically monitor those in images in real time. In these sort of few seconds to minutes time scales, we can monitor those changes. So we'd like to survey various particle sizes and surface functionalities. And we've started out by just using some simple tissue mimics like gels, and we've been moving on to cell packs and things like that. So this is just a sample T2 weighted image of contrast agent, which is in this outside. Okay, so basically there's a container and then a capillary with gel inside it. We're watching the contrast agent absorb into this gel over time. Okay, so there's not a whole lot of information here to convey, except that we can monitor this over time, and we're doing this with a range of different particles and also different tissue mimics. So we started with gels, we've been moving on to cell packs. We're interested in if people have engineered tissues that we might be able to access that would have, we're trying to understand, nobody's really made engineered tissues to engineer diffusion or flow properties. So we're interested in those kinds of things if you have that kind of expertise. And we have some various funding plans on the horizon for this next few months.